Hi, everyone. Thanks so much for joining. We'll be getting started momentarily. Welcome, everyone, to the second session of our series, Using Data Analysis to Understand ME/CFS and Long COVID. I'm Dr. Leslie Phillips with Solve, and I'll be your moderator for this session. Today, we're excited to welcome Dr. Jennifer Stone for session 2 of this series. Jennifer is one of the winners of our Stupski Awards, which support research using data from the U+Me Registry. I'd like to tell you more about the awards and the registry itself. First, a little about our webinars. After today's webinar, we will host a link to the recording, but during the webinar, we want you to know that as you ask questions, we hope to have time. It says here we will have time if time permits. We have planned time for question and answers, and those will follow the presentation. You can see on your screen how to get into the Q&A functions of the Zoom as well as closed captioning. And as noted, these webinars are recorded and you can find those recordings on YouTube. I am Dr. Leslie Phillips, but not that kind of doctor. I'm not a medical doctor, and I don't play one on Zoom, and none of us do. So if you do have questions for medical advice, we want you to go to your personal physician for that. Today, we'll be talking about the registry and the data from the registry. So first, I just want to introduce you, who may not be familiar with it, to the concept of the Ramsay Grants. These grants are intended to fund research and ME/CFS or long COVID. And the hope with these grants is attract new researchers to the field and keep them engaged. Also to facilitate applications. Often larger grants want preliminary studies, and it's sort of a vicious cycle of how do you get grant funding without the data? How do you get the data without the grant funding? And these are the kind of grants that we hope can facilitate those preliminary pilot data. And lastly, we always want to add to the cumulative scientific knowledge of ME/CFS and long COVID. The particular Stupski Data Analysis Awards are intended to fund projects that actually analyze the data from the U+Me registry, and they honor Joyce Stupski's generous support of research and her dedication to Solve. And Dr. Stone, who you'll hear from today, is one of the first 2 recipients. So let's talk a little bit about the registry, both for ME/CFS and long COVID. Here you see the ME/CFS enrollment. You can see the COVID cohort as well, and then the international cohorts. And this is sort of a bird's eye view, just to tell you how all those numbers break down across the registries. And then we also want to point out that one of the advantages of these registries is they can be agile. We don't just ask participants to offer data at one point in time, but we follow them over time to get longitudinal data. And what that means is that as science evolves or circumstances change, we can collect new data. An example of that is vaccination status data that we've recently collected. And here are some demographics specific to the ME/CFS cohort. You can see the age, sex, and functional status distribution. And here's that same slide for the COVID cohort. So I want to introduce our main speaker today, Jennifer recently received her doctorate in clinical epidemiology from Australian National University. She has already co-authored several journal articles using data and modeling to uncover causes of disease. Jennifer earned her BA and MPH from the University of Queensland. And today, Jennifer will be joined by Dr. Brett Lidbury. Dr. Lidbury's background includes molecular virology, and most recently, he has been leveraging machine learning pattern recognition techniques to support the study of human susceptibility to diseases to the disease of post-viral infection. You can send questions by clicking on the Q&A link at the bottom of your Zoom screen. Thank you so much for joining us, Jennifer. I'll turn it over to you.
No worries, thank you very much. I will share my screen if you don't mind. Let's have a look. Sorry, I'm just trying to share my screen and then I'll be with you in a second.
No worries, take your time.
Okay, can you see the slides? Yes, excellent. So hello everyone. Our research is focused on unraveling whether ME and long COVID are the same condition or whether they're distinct conditions and whether there are subtypes within each. Yeah. And the research team is myself, Brett Lidbury, and Alice Richardson. You've just heard a little bit of my background. I'm currently working for JBI in Adelaide, Australia, and I'm very much focused on methodology related to meta-analysis, which is, it uses quantitative statistical models to collate and average the results of individual studies. So I don't usually work with participants directly, but with the results already obtained within studies. And I'm specifically interested in how we can account for bias in the results of meta-analysis. And when it comes to bias, I've been particularly focused on bias in observational study designs, which is the type of study design that we have with the Solve-ME data. So we also have Brett here today joining us, and it'd be really great if Brett could introduce himself as well.
Yes, good afternoon, everyone. Thank you, Jennifer, and thanks to our gracious hosts. I do want to say that very grateful for the funding and the recognition, particularly for Jennifer at this early stage of her career. It was touched on earlier, but I come from the laboratory. My background's actually in experimental science in the lab, where I spent a lot of time studying a virus, an Australian virus called Ross River, which in Australia is one of the main suspects for post-viral fatigue and, by extension, ME. So that, without going into the fullest, lengthiest story, basically was my start as a virologist that came into ME research. And as said in the kind introduction, I've leveraged that. Now I'm no longer in the lab. I now work in computational sciences, in silico, as we say, using data, hence my collaboration with Jennifer and with Alice, using data to find patterns in complex data sets using machine learning and other statistical tools to identify patterns which would hopefully be predictive and useful to understanding these horrible types of long-term fatigue-related diseases. So that's me. I currently, I'm only part-time actually at the ANU. I'm an associate professor with the National Centre for Epidemiology and Population Health. So thank you, Jennifer. Back over to you.
No worries. We also have Alice Richardson, who's a statistician, and we've actually added another person to the team named Scott Moritz from Radboud UMC. He's a statistical consultant in the Netherlands, and he's going to be helping us with a lot of the machine learning methods that we're using. Okay. So first and foremost is a very big thank you to the participants of the Solve-ME study. So without their commitment of time and effort and giving us access to their very personal lives, research like this wouldn't be possible. And so it's a huge commitment. The surveys are quite long and tedious, and the dedication participants show to finding answers to some of the big problems in the field of ME and long COVID is very much appreciated. Also, thank you to Solve-ME and the Ramsay and Stupski grant schemes, as these are what brings participants and researchers together to investigate these conditions further. And so before we get started, I would be really interested to hear who in the audience has ME or long COVID. If you could just answer one of the boxes and we'll see what comes up. I'll give you a couple minutes.
I think we can close the poll now.
Oh, that's really interesting. So we've got almost 70% of people in the audience who have a diagnosis of ME, and 15% have long COVID, and 8% of people in the audience are actually caring for somebody with ME or long COVID, and another 8% have neither. So that's really interesting, and I hope that everyone's able to get something out of the presentation today. So we have 3 research questions that we would like to investigate. The first is whether COVID-19 is an independent risk factor for ME, as whether it predicts ME once we control for everything else. The second research question is what are the similarities and differences between individuals with ME and long COVID with respect to their demographic profile and clinical characteristics. And thirdly, we're interested to see whether ME and long COVID are just single syndromes on their own, or whether they have subtypes conditions have subtypes, and we're interested to see what kind of subtypes might come out of this data. Just need to, move this panel out the way. Yep. And so I am personally interested in a few translation questions. How this knowledge will add to what we already know about ME and long COVID conditions, and what needs to change based on the conclusions of the research. And then I would like to try and go a step further and look at how do we actually implement these changes, because that is probably the most difficult part of all of this. And so in terms of our research questions, and our research methods, we've started looking at research question 3, and this is investigating whether there are subtypes of ME and long COVID. And so we could categorize participants based on predefined subgroups. So if participants had mainly neurological symptoms, or mainly psychological symptoms, or mainly gastrointestinal symptoms, we could put them into subgroups based on their main symptom type and see if these made reasonable subtypes. But instead, we felt it would be better to throw our data into the system and see how the participants group together based on what we call an unsupervised process, so no input from us. And so to do this, we would use a dendrogram. And so we throw it all in, all the responses to the symptom questionnaires and the demographic information, and we see what pops out. And so as you can see, and this is just an example off the internet, this isn't the actual data, but all participants are down the bottom of the dendrogram, and subgroups start very specific and they become more broad as you move towards the top. So researchers can make a decision where the cutoff will be based on what's reasonable and what the data looks like. And you can see in the example on the right here that they've got 4 subgroups. One is a blue, a green, an orange, and a red subgroup. And so this represents 4 different subtypes of that particular condition. And so this is the methodology that we will use for this particular question. And we're still debating how best to address some of the other questions based on how the data looks. So I have another question for the audience. I think it's very important and interesting to know what the time was between when you first experienced symptoms and when you received your diagnosis. And I'll just let you answer that for a moment. Oh, interesting. So 27% have said less than 1 year. 14% said 1 to 5 years. 14% also said 5 to 10 years, and 23% over 10 years it took to get a diagnosis. And another 23% haven't even been diagnosed yet. So that's a really interesting result. And there are many ways that you can diagnose ME, and a lot of people are not diagnosed, which leads us into some of the problems that we have with this kind of research. So there are many different clinical classifications of ME with no real consensus on which is the correct diagnostic criteria. And so depending on how we select participants into the analyses, whether we make a stringent criteria, means that results might differ depending on that classification. And so we have a cohort of participants, and some are self-diagnosed and others are physician-diagnosed, and some meet some of the classifications for ME and others don't. And so as researchers, we have to make a decision whether to include all of the participants, regardless of how they classify, or how stringent to make the inclusion of participants into the analysis. And so the diagrams I have here, they are epidemiology diagrams, so try not to freak out, but these are called directed acyclic graphs, and they show us the relationships between variables in a study. And so on the left, what we're seeing is selection bias. Some people call this collider bias, where depending on the participants we select or exclude, we may get a different result than what is true in the population. So basically, we could get a result that shows an association when there isn't one, or we could get a result that shows a larger or a smaller association than what is actually true. And so on the right, you can see how this works. So we can see that either having a dead battery or having no fuel causes a broken down car. And so we can gather that if we have a dead battery and the car's broken down, then it's probably not an issue with the fuel. And if we have no fuel, we don't really have an issue with a dead battery, right? So what we're describing is a negative or inverse association between dead battery and fuel, such that when one is high, the other is low, or when one is causing a broken down car, the other one is not. And so we know that this association is not present in real life. There's no association between having a dead battery and having no fuel in your car. And the reason this association is present here is because we have selected only broken down cars into the analysis. So when we select just broken down cars, we get this association, and how we would avoid this problem is to select all cars, not just broken down cars, and we would see this association disappear. And so when we take all the participants that we've been given and we try to diagnose them according to different, more stringent diagnostic criteria, we can see the numbers drop. And so we have 3,000 odd in the ME group, 845 in the long COVID group, and 465 in the control group. So that's, about 4,500 participants in the study. And then when we diagnose them according to the international consensus criteria, we then end up with 872 in the ME group and 166 in the long COVID group. So a total of about 1,000, 1,200 in the entire data set there. And so this is not just a problem of more stringent criteria, but it's also an issue of missing data. So when participants skip over questions, we can't always classify participants, and we risk losing them from the analysis. And I'm very much aware that Solve ME have brought some statisticians on board to try and account for this and work out ways that they can keep these participants in the data set and work out ways around this and ways to keep using that very valuable data. But you can see using the ICC criteria, we do lose roughly 2,000 participants from the ME cohort. And so this is just the other diagnostic criteria, so that I wasn't just focusing in on one, but you can see roughly the same numbers. There's less numbers with the Canadian consensus criteria, more numbers with the CDC and with the ICC. And so this is the Karnofsky Performance Scale. It's a tool for functional impairment, and it's used to improve understanding of participants' ability to carry out daily activities. And so these are the score cutoffs, really. If you have a very low score, you're quite severely ill, quite severely impaired, I should say. And if you have a very high score, you don't really have, it's not really impacting on your day-to-day activities as much. And so these are the scores for the whole data set for ME, long COVID and controls. And you can see that there's 1,600 odd in the ME group with no complaints or no functional impairments, and 300 odd in the long COVID group that have no complaints. And then we see a sort of clustering around the 70-60 mark, which is requiring assistance and unable to carry on normal activities. And then we see, again, around the 30-40 mark, more participants disabled and severely disabled by their illness. And this is the this is how the Karnofsky scores look based on those who are in the who are diagnosed with the Canadian consensus criteria. So you can see that there's 196 with no complaints. You can again see the similar patterns around the 70-60 mark and around the 40-30 severely disabled mark. And so that's the CDC criteria, again, a similar pattern, regardless of the drop in the numbers. So that's good, I think. But how we would overcome some of the problems that I've just described with selection bias is to conduct what we call sensitivity analyses to check the assumptions of the study, specifically the assumptions related to selection of participants into the analyses. And so basically what we would do is run the same analyses on different, using increasingly stringent criteria. And if we get a really we get the same result regardless of the participants we use, then we are confident that this result holds true. And if we get different results, depending on how stringent we are, then we may have an issue of selection bias. And in that case, what we can do is just report all of the results and describe what we think is going on with that. But as I said, SolveME have also got some statisticians on board to see how we can use that data more effectively. So the impact that we're hoping that this research has on participants and on future research is that we're hoping that the subtypes that we come up with will help to inform the current diagnostic criteria. We're also hoping that the long COVID research, since it's such a big thing right now, will help to inform ME further as well in terms of diagnostic criteria, potential treatments and other such things. We're really hoping to spotlight attention on ME and long COVID conditions to increase funding in this area, because with funding, you get better patient outcomes. And I think that's been shown across the board for many, many different conditions. And I think I would like to continue in the area of ME and long COVID and look at research in the future, perhaps in the area of evidence synthesis, which is my area of expertise, collating all the data out there already and looking at whether it's consistent and whether we can form an international guidelines panel and start to get some consensus in this area. And that is all I have. Thank you very much for listening. And that's my email address. If you don't have time for questions today or something pops up later, you're welcome to email me and ask any questions about the analyses or anything. And that's my Twitter handle as well. So yeah, over to you, Leslie.
Thank you so much for a lovely presentation and such a thoughtful approach to these data. I am going to open it up to questions, but I'm lucky, I'm the moderator, so I'm going to ask the first question. You know, as a scientist who's comparing, you know, thinking about ME/CFS and long COVID, that's one thing, but we often see this in the media, you know, the kind of the compare and contrast between long COVID and ME/CFS. I wonder, you know, what your take on that is, if you have any comments.
Well, I think the biggest issue is that I don't see that in the media. I see a lot of stuff on long COVID and no link to ME/CFS. And this is something I've discussed with Brett as well. We recently had a lady who had given birth recently and was diagnosed with COVID during her pregnancy. She had, you know, joint pain and other such things and was describing this long COVID condition, and no information about ME, no mention of it in the news at all. And so I think that's quite interesting, and maybe Brett has more to add.
Yeah, thank you, Jennifer. And look, I must confess to being quite irritated with some media reporting. Now, through various sources, I can see reporting from across at least the English-speaking world, and there are some who are saying, oh, look at long COVID, it's a brand new disease. And of course, that upsets me. There's a lot to be learned. To my view, and I don't think it's controversial, is that long COVID is ME induced by the COVID virus. And why do I say that? Is, I mean, my understanding is ME particularly can be triggered by other negative stimuli. There's been some talk about heavy metal toxicity and other stressors, but there is, I think, among the scientific community interested in post-viral fatigues, well, that says it all, that, yeah, ME is a type of virally induced long-term fatigue, and COVID-19 is just the latest addition to the list of viruses that can be responsible for this. And the media reporting, look, I have picked up, like through our local Australian Broadcasting Commission, the ABC, I have picked up some really nice articles more recently that have actually linked the two together. But there is also, as Jennifer suggested, reporting to sort of say that, you know, that there's just no mention of ME, which I find disappointing.
Well said. Thank you both. We have a question, and I do want to let everyone know that Dr. Stone is at, really the early part of the analysis of these data. So I'm guessing no conclusions there, but maybe any insights, early ideas, anything a priori, or perhaps, why you chose to go with an unsupervised machine learning model, what the alternatives are. I'll kick it over to you to discuss anything you'd like to about your approach and your research and insights.
No conclusions yet. I do hope to do another presentation when we have some more results. Nothing that I would feel confident in telling you about now before we've sort of checked the data and done things like sensitivity analyses, because it could be quite misleading. But no, it's quite an interesting data set, and I think the qualitative responses as well are quite interesting, and we do plan to look into those with some machine learning as well. Why we chose an unsupervised approach, I think it's going to be more informative to have to be seeing what these subtypes are, irrespective of what our subjective input could be. And so I think it's a more objective approach to looking into subtypes of this illness. Perhaps Brett might like to. I don't know if Brett's got any.
No, I think it's a good first step to look at the structure of the data. In fact, on another project, I've recently used cluster analyses just to sort of, I suppose, break a really big, what at first glance looks amorphous set of data into sort of digestible chunks, if I can put it that way. So I think it's a natural place to start. And of course, once Jennifer and the team, and again, Jennifer's captain of this team, I'm one of her loyal lieutenants. Once we actually, those questions about clinical criteria, to me, that's a huge one, about getting some consensus. I don't think researchers and clinicians are going to change overnight and suddenly all agree to use Canadian or Fukuda or the international, whatever it might be. But coming from a reverse angle, trying to find maybe what brings those criteria together and what we can draw from that is probably a quicker way to go. And once we have a better idea, we could then do some supervised models. Once we have responses or we have criteria by, what's severe, what's moderate, etc. What sort of things we could then look at as meaningful responses, which we could then model.
I wonder if we could explain for the audience just basics of machine learning and what it means to take a supervised or unsupervised approach. Yeah, I know sure. That could be a whole webinar, but just brief.
There's also a danger of asking academics to explain things like that because we just go on. And I've just had my first coffee, so don't watch out. No, you happy for me to handle that, Jennifer?
Yeah, that's fine. I think it's a better place to do that. Yeah.
Yeah. Well, in a nutshell, basically with machine learning, these are algorithms which you hear so much about these days. So they're sort of basically formulae which basically look at data and try and separate it in a usually, a higher dimensional space. But anyway, that's inside the machine. But what it means is a supervised model is one where you're looking to predict or understand a response. So a response might be ME, positive, like someone diagnosed with ME, healthy, or non-ME, for example. That's a response. That's an outcome you could then model through a supervised machine learning model in looking to understand what makes someone ME or not ME. Whereas an unsupervised model, you're not asking or putting any assumptions about a type of response. It's just basically a model which, as I said earlier, just pulls apart the data based on means and medians and other statistical measures to actually then give us an idea how the data is structured without actually any reference to any other sort of conditions.
Thank you. I think that's an excellent answer and so much more enlightening than the YouTube I watched on teacher bots and learner bots. So much appreciated.
Pleasure.
A question about has SOLVE-ME thought about making data more public to encourage more analyses, particularly by folks who may not be associated with universities and the like? I promise we did not plant this question, but it is so well timed because the answer is yes. And very soon you will see on our website a link for a research form, which is not just for researchers per se, but anyone who is interested in using these data and ask a series of questions about the data use and the hypotheses, and if that person might have experience using data like this before or no experience, and all are welcomed. Another question, would it be accurate to equate environmental toxins and post-viral triggers to the dead battery and no fuel causes? Trying to understand the broken car metaphor as it relates to ME/CFS.
That's a difficult one because I need a drawing board to work out the relationships actually. What I showed you was a very simple example, and you can have parents of variables that influence other variables further down the line. And these drawings can become very complex with errors all over the place, with confounding and selection bias and other such things. I'm not sure about environmental toxins. I don't know if Brett can shed any light on that, but it's definitely something that if you wanted to flick it to me in an email, perhaps I could draw something up, but on the spot, it's a little bit difficult.
Yeah, in the ME context, the battery, or in American English, battery that we refer to, is the mitochondria within the individual cells. And actually, if we have time, I do have a slide, but I won't show it now. But basically, with collaborators in Australia, particularly Paul Fisher's group at La Trobe Uni in Melbourne, they actually found that there are some mitochondrial dysfunction in cells from ME patients that have joined our research programs. So the battery metaphor is a excellent one for that reason. There is, with our evidence in the laboratory, problems with the function of the battery. The fuel, I mean, the analogy or the metaphor that Jennifer used was absolutely spot on, because, you know, is it what's coming into your system which provides you with energy, or is how it's utilised? It's a problem. So the broken down car is perfect for that, to understand it.
Thank you. Another question from Greg. Is there any familial clustering in long COVID or ME/CFS?
I haven't actually looked into that yet. That's one of the demographics that we have listed in our analysis plan that we submitted to SOLVE-ME. Definitely interested in looking at family history, as well as how the subtypes map back onto the diagnostic criteria and other things like family history and demographic variables. I'm not sure if Brett can shed any more light on that based on other research?
Yeah, look, I've been involved in ME research for much longer than Jennifer. I've been working in this area for just over 10 years now. So just by exposure, I have some anecdotal answers to that question. And as Jennifer suggested, nothing firm driven by data, but I'm aware of families where there have been cases of ME through the generations, but also autism. But that's just completely anecdotal, no evidence. It's just basically what I've seen just through my experience talking to people. But in terms of the science, there's, to my knowledge, no link established in terms of it being a genetic disorder. But I would say that mentioning things like autism, for example, which are very complex, there is a gene involvement, but there's some environmental factor as well which interacts, which of course could be a virus, or in many cases will be a virus. So the family histories are there, but to my knowledge aren't very solid at the moment, and again haven't been, I think, properly studied in Australia anyway. I'm not sure about elsewhere.
I know we had a discussion about mitochondrial dysfunction, and I think that slide is lovely if you want to go ahead and share it with the group.
Yeah, sure, Leslie. Just give me a moment.
Yeah, I know it's always hard to get into the share function, so take your time, please.
Yeah, it's always a juggle. But actually, just while I'm doing that, Leslie and Jennifer, I did actually respond to a question from Virginia too about biomarkers, and provided what I'm sure is a very unsatisfactory answer, and one that is very typical of academics about funding. But yeah, I feel a big barrier is access to the funding to do the proper large validation studies. I mean, there are many candidates, biomarker candidates for ME, but to take it to I suppose a larger level within a population to study it with thousands of participants and across many centres is a very expensive undertaking. So that's, I think, on the ground, one of the big problems we have with taking some of these great discoveries forward to actual, for the benefit of patients in terms of diagnoses. So, all right, let's have a try.
And as, yeah, oh, perfect.
Yeah. So yeah, just wanted to say, now I will, oh, yeah, I have trouble. I want to switch to the full presentation, but I won't risk that. So yeah, I have to give homage to my colleagues at La Trobe Uni, who have done a lot of work on immortalised lymphoblasts from ME and what we call control patients. And we're starting to talk about control patients as not healthy controls, but as non-ME patients. And we have, as I mentioned earlier, some excellent cohorts based in and around Melbourne in the southern part of Australia. So this is the diagram. So yeah, hang on, put on your seatbelt for this one. But essentially what it's showing is, if I could just make that larger, I'll do that. Again, there's, so basically what you've got is the inside and the outside of the mitochondria. And of course, inside is where you've probably heard of ATP, which is the currency or the fuel that we use. And essentially what Paul and his team has found is that there's I suppose what you'd call a lesion or inefficient ATP synthesis. So the basic energy, like the little, I suppose we call it the currency. If you think of dollars or coins as something that needs to be given and taken, you know, to provide energy, in complex 5, which is embedded in the mitochondrial membrane, there's been some problems found with how that functions to provide energy. And of course, if there's a problem with energy production, there's a problem physiologically and therefore for the individual concerned, in that they can't, you know, live their life. They don't have the energy to perform day-to-day tasks. One thing, again, anecdotally, with my experience, both my own direct experience and working with colleagues, is of course post-insertional malaise, which I find is really, really central to ME and the experience of ME. A lot of people say, well, there's a lot of conditions, human diseases and disorders with fatigue as a symptom, but this post-exertional malaise makes a lot of sense in terms of these mitochondrial studies, because you have energy available, but once you use it, it's not replenished efficiently. And this mitochondrial function work that's been so brilliantly done by my colleagues is, I'm sure, part of the answer. I'll just show this as well. This is associated with, again, being a typical academic, I can't help myself but to talk about a publication. I did mention Ross River before, which is an Australian virus associated with post-viral fatigue and ME. And basically, where I'm arriving theoretically, which links together the mitochondrial work with other facets of ME, is what we call homeostasis and the loss of it due to an infection or some other insult that leads to chronic long-term fatigue. And homeostasis is just basically a fancy Greek word for equilibrium, which is a French word, but anyway, in English, equilibrium or your balance. If you just think of balance in your system. So what we're looking at is a loss of balance. And what I proposed in this article was there's a virally mediated disruption. And it seems, again, from the work done by Paul and others, disruption of this thing called TORC, which is a full name is a target of rapamycin. It's a type of protein which has been found to be really, really important in terms of maintaining homeostasis. And of course, if that is disrupted, as shown here, that's basically the control group, but you can see there's a hyperactivation of this TORC. So this balancing protein is increased significantly, much more significantly than if you just look at people without ME. So this loss of homeostasis, activin's another potential biomarker we've worked on, which is proving difficult to validate, but nonetheless, activin as a protein interacts with TORC. So this is purely theoretical at this stage.
Thank you so much. I know that is a lot, and we did have to put our seatbelts on. And in fact, that next survey question is going to be about activin and TORC. So I hope you were listening carefully. No, I'm just kidding. But I think it's always good to see some of the science that's going on in the background. Another question from Carrie, which I'm going to comment on and then turn it over to our presenters, which is sort of relating to a previous question. Can these data be, that the data that are being collected persuade funding entities to realize this impact when we take it and we look at new research data that's very new with long COVID and we combine it to ME CFS? And I will say that's the hope, that's the holy grail. That's why we're doing this work. Private entities can only get so far. There are big grants out there that come from government entities, and that funding needs to be coming the way of ME CFS and long COVID as well. And that sort of kind of leads to a question I had, which is, how do you think that learning about similarities and differences in the connections between long COVID and ME CFS can help move the field forward?
Well, I think the lack of consensus in the field is really holding everybody back. And I think if we can, this will only be one study, and there needs to be lots of studies showing sort of the same thing, if possible, to really, I think, make a change. But I think looking at the similarities and differences between long COVID and ME, will, I guess, in terms of a post-viral illness, give more weight to that theory, I think, yeah, and that will really, yeah, I think that's probably the best hope we've got with that one. What do you think, Brett?
Yeah, among my colleagues, and I work with people in the UK as well, so it seems to be consensus across a few places, is that, with all things, there's politics, and I'm not sure about the United States, but in Australia, I know certainly the UK and the Netherlands, there's a problem with ME. There's still, unfortunately, in the medical profession, a lot of ignorance by some members of that profession, and as Jennifer said quite correctly, it's led to a fragmented type of, I suppose, approach to ME. There's still people talking about graded exercise and CBT, and even though the evidence has built over the years to challenge that quite strongly. So I agree with what Jennifer says, that it's sad that we had to go this way, but long COVID might just be the fillip that we need to actually get people to take ME seriously. And in fact, among that group of colleagues I mentioned, just again anecdotally, and not in terms of publication or any official sort of statements, is that just in fact to handle the politics and to try and elevate all of these questions around ME, we need to start talking about post-viral fatigues in a more generic way, which people seem the medical professionals, I don't know, but people generally seem more, particularly in light of long COVID, seem more comfortable with. The history of ME, unfortunately, has become fragmented and made it very difficult. And again, in Australia, like the US, the UK and elsewhere, we face huge difficulties in attracting funding to do our work, which is related to that very problem.
I heard maybe some underlying concerns about the medical profession. I wonder if you can talk a little bit about that. Is that lack of recognizing the syndrome, dismissiveness, all of the above, something else?
Yeah, now I'm not talking about every member. Of course, there are some wonderful, medically qualified, medical practitioners, and we have a handful of champions in Australia who are talking to their colleagues, and we have a great advocacy body called Emerge, who's working closely with clinicians as well. But historically speaking, it goes back some years. There's again I suppose a cultural element. I don't know if you in the US used to have the majority yuppie flu, and there's sort of this view that there was just a malingerer's excuse. There's that. So the sort of bad press, and again, the media historically have done a very poor job in highlighting those sorts of comments and stigmatizing people. That's improved in Australia, I'm happy to say, and there's been some very powerful stories reported in more recent times to balance that view. But at a more scientific biomedical level, the other problem we have, and this is something we're actively working on, is a person presents to a general practitioner in a suburban or a town medical practice. They come in and describe the symptoms that we are aware of with ME. So the usual step in Australia is there'll be a number of tests. So blood will be drawn and sent to a laboratory. And what happens is all the laboratory results come back as within the reference interval or in the normal range. And then the general practitioner, who's not a specialist, but then just follows basically a set of guidelines and says, well, either it's in your head, you need CBT, or I've heard a bit about graded exercise therapy. And our guidelines haven't been updated in Australia since 2002. So that's a huge problem. And we can't expect GPs just to instantly become experts overnight. So they unfortunately tend to just sort of say, well, the pathology and the tests that we've run show nothing abnormal, therefore there's nothing there, or you're imagining it. And unfortunately, I have heard stories. I do talk to patients and advocates a lot. There have been, of course, cases where some members of the medical profession can, sort of intimidate or patronize or humiliate people as well, which makes the situation worse. I'm not saying that's the rule, but it does happen. And of course, we do, as even with scientists and researchers, we talk to patients and hear these stories, which are quite distressing.
Thank you. Well, unfortunately, we're running toward the end of today's webinar. I just want to let all attendees know that you'll receive an email with a link to the recording of today's session, and we'll also post it to social media as well as to our website within 24 hours. We've also shared a link to previous recordings and webinars in our website, and that will be in the chat as well. And I just want to say thank you so much to Brett and Jennifer for joining us today. It is so exciting that even just at the beginning of your work with our data, you have so many insights to share. And we hope you might agree to join us another afternoon/morning for you when you get to the end of your analysis. We sure appreciate having you today. And thanks to everyone who attended and for your fabulous questions and participation and engagement. We really appreciate it.
Thank you.